Hello, and thank you for joining us today in the next webinar of the Jasco webinar series, Alternate Detection Techniques. And what I mean by alternate is alternate to the most commonly used UV-Vis PDA that you see in a lot of labs today. So here's what I'm going to talk about today. First, we're going to start off with a Jasco overview. We'll take a look at how Jasco started, the products they have to offer, and then a short section on Hplc versus SFC detection. This will become important in the subsequent slides, as I'm just really going to illustrate what is different about a detector that is used in Hplc versus what is different in a detector used by SFC. And then all the subsequent slides, I'll explain if the detector can be used in one or both. And then we're going to sort of follow the same scheme for each detector. I'll introduce the detector with the detection summary. We'll take a look at the operating principles, and then we'll follow up with some applications. So the first detector is circular dichroism. The second detector that we'll look at is refractive index. Next up, optical rotation, fluorescence detection, and then finally, evaporative light scattering detection. So to start, Jasco was founded in 1958. It was established at the Optical Research Institute in Tokyo. Its founding members include Dr. Tominaga, who shared the Nobel Prize in 1965 with Richard Feynman. Then in 1972, Jasco took their optical expertise to the Hplc product line and sort of built a system around it. Coincidentally, that was the same year that Jasco was established in the US. So here are some of the products of all the instruments that Jasco has to offer. We have CD polarimetry, fluorescence, UV-Vis near IR, vibrational circular dichroism, confocal Raman, FTIR and FTIR microscopy, SPM, and then finally Hplc and SFC for the chromatographic line. As far as the Hplc and SFC is concerned, all formats are offered. So we have UHPLC, semi-micro, standard HPLC, preparative, and same for SFC. So what we're going to do is kind of focus in on this area for the moment, just to explain some of the differences that you'll see between the two techniques. So first up, Hplc. Many of you are probably familiar with this. So you start off with a pump that can pump, up to 1 or 4 solvents. We're going to come down and mix the 2 solvents together, and then head off to an autosampler, into a column oven, then into a detector. And downstream of the detector, you can either go to waste. If you're doing preparative chromatography, go into the fraction collector, or some sort of destructive detector technique like mass spec. But it's important to note here that when you come out of the column in an Hplc, you are on the low pressure side of the system. So this detector does not see much pressure. In fact, most Hplc detectors flow cells are rated for anywhere from 600 to 1,000 psi. SFC sort of introduces a few new modules, and there's some differences. So first, you start off with a CO2 supply. So instead of a solvent, in pump A, you would start off with a CO2 supply. This is liquid CO2. We chill it. We bring it into the pump heads chilled to make sure its density is as close to 1 as we can get it. This way, we are actually pumping what we say we pump, because CO2 at higher temperatures the density will vary greatly. Then we're going to mix in some sort of co-solvent, usually an alcohol with SFC, just something with a far different polarity than CO2. This way you can mix up you can increase the diversity of the compounds that you go after. So just like Hplc, you're going to mix the 2. We're going to go into an autosampler, a column, and then into the detector. But just downstream of the detector is really the major difference between these 2 techniques, is what we call the back pressure regulator. Now, I mentioned in Hplc, when you come out of the column, you're on the low pressure side of the system. If we were to do that in SFC, basically CO2 would expand at a 500-to-1 rate. It would cool very rapidly, and you would never get a signal in your detector. So that's what the job of the back pressure regulator is to keep pressure on the system throughout the flow cell of the detector. So that means the flow cells in the detector need to have a higher pressure rating. Typical operating pressures for an SFC are anywhere from 100 to 400 bar. So you at least need to cover that range and what that flow cell can withstand. So first up, circular dichroism. This detector can be used for both HPLC and SFC. And as you know from the previous slide, it's a way of saying a high pressure flow cell has been created for this detector. So the detection summary. So circular dichroism detection offers high sensitivity for chiral compounds. And if you're not familiar with the chiral world, chiral compounds are compounds that have one or more chiral centers and are mirror images of each other. These mirror images are known as enantiomers. When a chiral compound is synthesized, it undergoes a process known as racemization, which is the creation of the second enantiomer. Back in the 70s, not much was known about this process when a drug called thalidomide was created. The enantiomer of interest was synthesized and it worked well for its intended use, which was to treat morning sickness. Unfortunately, the mirror image compound that was created through racemization had side effects that included birth defects, which was a really unfortunate combination of two side effects. After this, the FDA mandated chiral testing, and companies then had to figure out how to address this. So you can address the racemization one of two ways. You can let the racemization take place and then use preparative chromatography to remove the unwanted compound, or you perform a stereosynthesis to produce only one. Typically, one of these methods is more cost-effective, and which one depends on the compound, but either way you look at it, the resulting batch will need to be tested for enantiomeric excess, or EE. So chiral compounds absorb to a different extent for left and right circularly polarized light, and the detection is based on the difference between the left and right hand circular polarizations. This technique is insensitive to changing temperature and solvent conditions, which is another way of saying you can run gradients with this detector. And Jasco is the only one to offer circular dichroism in the chromatography. So the operating principle, you start off with a mercury xenon lamp. The light is emitted by a light source and is collimated by the lens and introduced to the polarizer where the light is linear polarized. The light is then dispersed off the diffraction grating and directed to pass through the photoelastic modulator, or the PEM, which is really the heart and soul of this detector. The PEM then modulates the light to left and right circularly polarized light, and it changes back and forth really quickly. The modulated light is directed to the flow cell by the lens and then onto the detector itself. So here's a diagram of time variation light intensities. So the top is basically an example of what you would see on a chromatogram, so signal versus time, when no CD active compound is in a flow cell. So what you see is the light changing directions at fast intervals, but you don't see any signal. Down here, this is an example of when a CD active compound is in the flow cell. So you see the change in the direction of the light, and what you see is corresponding negative and positive peaks on the chromatogram. So that's really the biggest difference you would see between, say, this and a UV detector when looking at your compounds. One peak is going to be negative and one peak is going to be positive. So what does this mean to the chromatographer? So this is the chiral separation of flavonone and their corresponding CD spectra. So what you see here, when you go to develop a method on a CD detector, you're going to do it in a very similar way that you would do it for a PDA detector. So you're going to run your spectra, you're going to look at the wavelength maximas, and then you're going to test each wavelength where you see the best response. So here, the wavelengths that were chosen for this particular compound were 250, 310, 340, and 370. So you can see at 250 and 310, we have a negative peak and a positive peak coming out. negative peak and a positive peak. But what happens after 310 is they switch selectivities. And now what we see is a positive peak and a negative peak. So this could be real problematic for somebody using a UV detector in this instance without individual standards for each enantiomer. You could very well go ahead and prep out the wrong material and not even know it until it's too late. But the real power in this detector comes from G factor calculation, or what we call differential absorbance. So G factor is the change in absorbance units by the CD divided by the absorbance value of the UV. G factor is proportional to optical purity and is independent of concentration. But the take-home message here is that G factor trace indicates areas of optical purity on the chromatogram. So simultaneous CD and UV detection allows you to pull out as much of the material you can without having much crossover, without complete chiral separation. So here's an example of that. So what you see here is the blue trace. This is an incomplete separation of ibuprofen. And the blue trace represents the UV trace. The black trace represents the CD trace. And the red trace represents the G factor. So if you were to look at either the UV detector trace or the CD, or the CD detector trace and try to determine where to cut these peaks in order to get maximum recovery and maximum purity, it would be pretty difficult to predict. But the G factor offers a flat area in a flat chromatogram in the optically pure area. So if you take a look at this, the start and stop times of the collection become very apparent. So here's an example of a reanalyzation of fractions triggered by UV and then also triggered by G factor. So on the left-hand side, we have the UV trace. So these start and stop times were picked manually. And if you look below here, you can see, and that was based on the UV chromatogram. If you look down here, fraction 1 was 96% pure, and fraction 2 was 92% pure. But then we went ahead and applied the G factor, and we changed our cuts to fit the optically pure areas, and wound up with a fraction number 1 at 99.8% and fraction 2 at 99.6% without sacrificing much recovery. So some example applications are chiral samples that contain a chromophore. So it's an important distinction here, because as we get to some other detectors, you'll see that not everything requires a chromophore. But these are chiral samples that contain a chromophore, so drug compounds are probably the most popular use for this. Then chiral pesticides and chiral amino acids with derivatization are two other examples. So here we see the Hplc CD detection of amino acids. This is pre-column derivatization with DAB-CL. Here you see a chromatogram of proline, serine, 309, cysteine amino acids from top to bottom. And as you can see, each one has a corresponding negative peak and a positive peak. This is the Hplc detection of fluoroprofen. So again, now comparing it to the UV separation, you can see you have the negative peak and positive peak versus the two positive peaks. All right, so on to refractive index. Now this detector is Hplc only, so there does not exist a high pressure flow cell for this detector. But that's not the only reason we can't use it with SFC. So the detection summary: when light exits one type of medium and enters another, it bends or refracts. So RI's detectors measure the change in the refractive index of the column effluent passing through the flow cell and the compounds being analyzed. The greater the difference between the refractive index of the mobile phase and the refractive index of the compound, the larger the RI signal. So as long as a compound has a different RI than the mobile phase, it can be detected, which makes this technique very universal. The downside is it's not quite as sensitive as other detection techniques that exist, and it's typically used when you don't have a whole lot of compound, when you have plenty of compound to work with, sorry about that, and low-level detection is not needed. So the detection relies on the background being constant, so gradient elution is not recommended, which also brings us to the SFC point. So a supercritical fluid behaves very differently than standard liquids you would use in the HPLC. So a supercritical fluid under a certain pressure and temperature, the RI will vary greatly with minor variations in pressure and temperature. So if you tried to use this on an SFC system, you would have a hard time with the background and therefore probably drowned out your compound signal. So the operating principle, the operating principle is based on the angle of deflection, which is proportional to the difference between the refractive index of the solvent and the refractive index of the sample. You can see the variables we have listed over here. So we have the refractive index of the sample, we have the refractive index of the mobile phase, the angle of incidence here and here, and the angle of refraction. Now, these all go into an equation and produce the angle of deflection. And you can see the relationship right here. So some example applications, samples that lack a chromophore, so inorganic ions, GPC, SSC applications, sugars. Here's an example of chondroitin sulfate by SEC. The top, you see the molecular weight distribution standards. This is a Pahlulan standard mix. And below that, you see the quantitation of the chondroitin sulfate C sodium salt. Here's a sugar alcohol analysis. So here you see glycerin, erythritol, xylitol, sorbitol 5, and inositol. Now, I mentioned that you cannot run gradients with this technique. So what are some other ways we can go about increasing efficiency? Well, recycling is one option. Another option is to use a bunch of columns in series, which a lot of people do. But in this case, a bunch of columns would not be necessary. So here you see we have our system. So we have our pump, we have our auto sampler, our column and detector, just like you would in any other system. As you come out of the detector, you come to a recycle valve. Now this recycle valve, when closed, the flow goes straight through. When open, it has two positions. You could either go to waste or onto a fraction collector. So for the first couple of cycles, you would then keep this valve closed. You would do your first pass on your column. You would do your first pass on your column, then you would come back up to a recycle kit and then head back to the pump into the auto sampler to a second pass on your column. And you would do as many passes as needed to get the resolution that is desired. So here's an example of that. You can see here, this was the first injection. We did not have much resolution. Move on to the first cycle. So this is the first go through the recycle valve, the second cycle, the third cycle, so on and so forth. So the object here is to take a look at the resolution after each cycle. And when you're not gaining any more efficiency, well, then it's time to collect. So we took a look at this fifth and sixth cycle, which looks pretty similar. And so we sent the sixth cycle off to the fraction collector. Okay, next up is optical rotation. This is also just an HPLC detector. And this sort of goes hand in hand with the two previous detectors, the CD detector and the RI. So the detection summary, this is optical rotation detectors also detect chiral compounds, very much like CD, but these particular compounds do not contain a chromophore. So the detection relies on the measurement of the angle of rotation of plane polarized light caused by optical isomers. The detector is sensitive to temperature and solvent changes, so just like RI, gradients are not recommended. The operating principle, again, you're going to start with a mercury xenon lamp. The light is emitted from the light source and is collimated and reflected by the mirror, The polarizer passes light of a specific polarization and blocks all others and sends it off to the Faraday cell. The linear polarized light enters the Faraday cell at a specific frequency and maximum rotation angle. The modulated light is focused on the inlet of the flow cell by lens 2 and passes through the cell and through lens 3. And then finally, the light then passes through the analyzer and is detected by the photodetector. So some example applications, I will say that this is not the most commonly used detector, but if you do have compounds that fit the bill, it works nicely. So I would consider it more of a niche detector. But samples that lack a chromophore, so amino acids, terpenes, sugars, and also contain optical rotation. So here you see the OR analysis of sugars. You can see at the top, this is the OR trace, and here's the RI trace. The top trace shows only the optical isomers, 2 and 3. But what we would have missed without RI detection is number 1, which shows up here. So these two detectors used in conjunction with each other can really serve a good purpose. On to fluorescence detection. Now, fluorescence detection can be used for both HPLC and SFC, and Jasco is the only one that offers it in the SFC realm. The detection summary. So when compounds have certain functional groups that can be excited by shorter wavelengths energy and emit longer wavelength radiation, this is known as fluorescence. So the fluorescence intensity depends on both excitation and emission wavelengths. The detection of any component depends on the chosen wavelength. So if one component can be detected at, say, 260 nanometers, or excited at 260 nanometers and emits at 340 nanometers, another component could be missed if you're not sitting on the correct excitation and emission for that particular component. So that's why modern detectors need to be able to sit on more than one set of conditions and also be able to switch conditions very rapidly. Some structural features of compounds that fluoresce typically fall into one of the following categories: conjugated pi electrons, aliphatic and alicyclic compounds with carbonyl groups, and highly conjugated double bonds. Only roughly about 15% of compounds have the natural ability to fluoresce, but if they do fluoresce, fluorescence detection is among the most sensitive type of detection for modern LC detectors. Even is to go as far as to say it's 10 to 1,000 times more sensitive than what you would see from your UV detector. So just to give you an idea of what kind of levels we're talking about, Jasco offers two fluorescence detectors for HPLCs and SFCs. This is our more sensitive one, the FP4020. This is Anthracene at 31.4 femtograms on column, achieved with a signal to noise of about 215. So the operating principle, you start with your light emitted from your xenon lamp, and it's dispersed by the excitation side diffraction grating and radiate towards the sample as an excitation light. A portion of that excitation light is split by the beam splitter and detected by the excitation side of the detector. From there, if the sample contained in the flow cell has the ability to fluoresce and is exposed to excitation light, it'll then emit a fluorescent light. And then from there, the fluorescent light is dispersed by the emission side diffraction grating and detected by the emission side detector, the photomultiplier tube. So some example applications are samples that require high sensitivity. So there are protein purification, protein stability, environmental analysis, food additives, in particular phenolic antioxidants, or tocopherols. So here you see the analysis of PAHs via fluorescence detection. and it looks like a pretty busy chromatogram. So very similar to that of CD or very similar to that of UV-Vis. You're going to start your method development process by scanning a bunch of excitation and emission wavelengths to see what compounds respond to what. Now, as you can see here, we have a whole host of conditions down here that are required to be able to detect every one of these compounds, so all 15 of them. So you can see fluorescence detection is much more specific than, say, a UV-Vis detector. So if you're working with a UV-Vis detector and you have some sort of ring structure or some type of chromophore, you can get a lot of those compounds with two wavelengths, 254 or 220. And that is not the case with fluorescence detection. It's much more specific, but as long as you have the ability to switch excitation and emission wavelengths really quickly, and you can monitor a couple conditions, you should be able to get everything that you need. So here's an analysis of PAHs and Anthracene, and this is fluorescence detection versus UV. Over here, this is the PAHs. You can see each one is on the same time scale, or the signal scale. This is the fluorescence, this is the UV. This is 2,500 picograms of each. And down here in the fluorescence chromatogram, you see this is 25 picograms of each. So about a 250x more sensitive. Then the Anthracene here, detection limits were calculated at 7,040 femtograms for the UV and 26 femtograms for the fluorescence. So as you can see, much more sensitive. All right, on to the final detector, which is evaporative light scattering detection, or ELSD. This can be used for both HPLC and SFC. So ELS detectors are mainly used for semi-volatile and non-volatile compounds. No chromophore is required for detection. Basically, as your mobile phase flows through the chromatographic system, both your solvent and solute from the column is nebulized and carried by a gas flow through an evaporation chamber. The solvent is then volatilized, leaving just your solute particles that scatter light. The higher the concentration of the solute, the more scattering that occurs. The signal is then amplified, and a voltage output provides the concentration of the solute particles passing through the light. This has very low limits of detection, down to about 10 nanograms, so not quite as sensitive as fluorescence, but very sensitive. And unlike RI and OR, gradients can be used with ELS detection. Compared to other detectors, it produces a more uniform response independent of physical and chemical properties. So really, this can be considered quite a useful universal detector. So the operating principle, your column effluent enters here and is hit by a nitrogen gas to induce nebulization. The nebulized solvent stream passes through an independently temperature-controlled drift tube where the solvent is removed, leaving just the solute. And then finally, the particles pass through the optical chamber and scatters light. The degree of scattering is dependent upon the concentration of the particles and also the size of the particles. So some example applications include samples that are semi-volatile to non-volatile, carbohydrates, lipids, polymers, non-ionic surfactants. Here you see an example of non-ionic surfactants. You see the molecular weight distribution produced for Triton X100 and PEG 400. This is cyclodextrins in green tea. So you can see all the matrix compounds that came out in the green tea and the various forms of the cyclodextrin. Here's a mixture of mono and disaccharides. So here you see rhamnose, fructose, glucose, sucrose, and maltose. I would say sugars are a very popular application for this and RI detection, and usually one or the other detector is used for that type of analysis. Here's analysis of fatty acids. So you can see the 9 fatty acids listed at the bottom here, all well resolved. So a summary. Circular dichroism is a good detector to help distinguish between enantiomers and assist with increasing enantiomeric purity with preparative chromatography. Refractive index detectors are universal detectors for samples without a chromophore. However, they're not quite as sensitive and gradient elution is not possible. OR detectors address chiral detection when a chromophore is not present and can be used in conjunction with RI detectors. And fluorescence detectors are the most sensitive detectors on the market. A low percentage of compounds can be detected with this technique. However, if a compound does fluoresce, it's a great detection scheme to employ. And then finally, ELS detectors are used for semi-volatile to non-volatile compounds, and their detection is independent of chemical or physical properties. So just a quick slide on some of our educational resources. First up, our upcoming webinars. Next up is Dr. Carlos Murillo. He is doing a live IR imaging demonstration from our lab, and this takes place on Tuesday, June 9th at 2 p.m. After that, we will have a presentation on UV fundamentals. We have ebooks, tips, and tricks posters for Raman, fluorescence, FTIR, and CD, and our knowledge base, which is really thousands of publications and applications involving our instrumentation. If you would like to contact Jasco about any of the things that you heard today in this webinar, please send your emails to sales@jascoinc.com. Give everybody a few seconds to write that down if you wish. And then lastly, I'll open it up to questions. So you can type your questions in the chat box, I believe, down at the bottom on the right-hand side.
Hi, hello, this is Dr. Carlos Murillo. I was supposed to introduce Tom at the beginning. Sorry, my phone was on mute. I just want to say thank you to everybody. Thank you to Tom. And we're starting with the questions. And I just passed 2 questions to you, Tom. They are popping up in the question window.
Okay. There we go. Hold on one second. I can barely see them. All right, I'm starting to see them come through now. All right, so first question. Does Jasco carry different format flow cells for their HPLC detectors? The answer to that is yes, for all applicable detectors. We have semi-micro flow cells for the UHPLC detectors, standard flow cells, and then also prep flow cells. The one exception of this is with the fluorescence detector, because you don't really need a more sensitive flow cell for fluorescence detection. One flow cell will suffice for all formats, and especially when you get to prep, basically you can just lower the gain on the detector so your detector is not overwhelmed. Okay, so you mentioned that the CD detector is only made by Jasco. Does that mean it can only be used with the Jasco system? No, we have many of our customers that use this detector on their adjuvant systems or water systems, whatever type of system you may have. With one side note here, so basically you can import the signal to most HPLC detectors that have that ability, but you will not be able to scan CD wavelengths. In order to do that, you have to have our software package as well as the detector. So you sort of take it offline, run our software, get the CD maxima, then plug back into your regular system software, use those wavelengths and carry on. And that's really true for all of our detectors. Can the Jasco fluorescence detector be used with the ACTIS system? Yes, we have many customers using these detectors with protein purification systems. And really, it's sort of the same thing as the CD detector. You just need as long as your system has the ability to import third-party detectors, you should be okay. All right, so for the CD detector, can you get a full CD spectrum when you click on the chromatograph? Yeah, I would say when you say, I guess it depends on what you mean by full CD spectrum. So this is not to be confused with a CD spectrometer. So you will not get the intricate spectra. You will not get the intricate spectra that you would get from a spectrometer. But yes, you can click on the chromatogram and at any time reveal the CD spectra as long as you perform the scan to do so. All right, that looks like about it. I want to thank all of you for joining us today. Like I said, if you want to contact us about anything you heard, please use the email address I showed on the previous slide. Thank you.
Okay, thank you, everyone. I just want to remind you that slides and the recording is going to be sent in within this week, and you are more than welcome for the coming webinars. Okay, thank you.